Hello folks, today we'll be talking about protein structure. And so it looks like there's a lot of learning outcomes today, but most of these are just definitions. So don't worry too much about the definitions, those should come fairly easy. Let's focus more on the deeper learning outcomes. So we should recognize that the primary structure of protein is simply the sequence of amino acids. So that's a definition, right? We should again recognize the secondary structure is the local spatial arrangement of the peptide backbone. Another definition. Now, getting a little bit deeper, let's recognize the three types of secondary structure and compare and contrast stabilizing forces in each. Back to a definition. Let's recognize the tertiary structure is the overall shape of a protein, including its side chains, and that there are many patterns of tertiary structure. 5, getting deeper, let's describe how protein folding occurs and how the hydrophobic effect contributes to folding instability. Let's compare and contrast domains and subunits. And the last little definition, we'll recognize that tertiary structure describes how more than one protein or subunit interact. Today's Meet a Scientist moment is Marie Maynard Daly, the first Black American woman to earn a PhD in chemistry in the United States. She made important contributions to the research of protein synthesis, among other things. For example, she helped describe the role of ribonucleoprotein in protein synthesis. And how she did this was kind of cool. She used radiolabeled amino acids, so radioactive amino acids, to determine how protein synthesis changed in fasting and feeding. Pretty cool stuff. Okay, why do we need to talk about proteins in the first place? Well, proteins perform nearly all molecular transformations critical to biology. Of course, not all. If you take a lot more biochemistry or molecular biology, you learn about how RNA can also behave as a catalyst sometimes. Proteins do a lot of cool stuff. They do transportation, they do mechanical work, they do electrical work, they do signaling, they regulate stuff. They do so much. Throughout this series of slides, you're going to see me kind of repeat this slide over and over because it shows the 4 levels of protein structure we will discuss. Primary and secondary structure, let's start there. Primary structure, simply the sequence of amino acids. What is their order and which ones are they? Secondary structure, the local spatial arrangement of the protein backbone. And here's a slide with your definitions. Let's jump into primary protein structure. So we covered this last time we were together with amino acids. We can form the primary structure of a protein by linking together amino acids to make residues within a peptide. Remember, we do this through condensation. We lose water across the bond to form a bond directly between the carbonyl carbon of one C terminus and the amine nitrogen of the N terminus of the next amino acids. There's a lot of diversity in protein because we have a lot of amino acids to choose from. We have 20 amino acids compared to nucleic acids that we'll talk about later, where we really only have 4 nucleotides to choose from. And proteins really do come in all sizes, right? In this table, why am I showing this to you? Well, just to show you that we have proteins as small as 30 residues, tiny. But we also have proteins as large as 35,000 residues. Some are tiny, some are huge. That is primary structure. Just the sequence of amino acids and how many there are. Next up, secondary structure and what is important to know about secondary structure.